in mind that these applications are completely relevant when you think about C. elegans and Drosophila. The same general principles apply. So how do we make sense of this so-called high content in vivo data? Again, first to identify the opportunities and again, full disclosure, some of the challenges in this approach and others. We all want to identify potential hazardous chemicals, and my lab works a lot with nanoparticles as well. We want to reduce the uncertainty, and we want to reduce this uncertainty with experimental data. We do want to do high throughput assays to fill these gaps, and like Chris mentioned, we're at the point of we actually can do high throughput screening at the numbers that he described in the true high throughput, but using whole animal systems. And again, what's limiting is really only facilities. It's really, there's no technical limitations to do high throughput testing in the whole animal, in zebrafish and Drosophila and C. elegans. We want to, again, we have the same goal. We want to develop predictive models, and we're trying to develop these models in-house in terms of our nanoparticle responses and also certainly with our chemical responses. But we also have to admit we need to be able to manage the limitations of this model. Again, what Chris mentioned, you really only get what you ask for. So there are limitations on the platforms that you develop, and there's limitations on the information you'll be able to extract from it. So this won't just be a promote the model talk, but there'll be some of that, of course. So we all recognize this kind of a linear thinking of toxicology, which starts from exposure and ends with a pathology or disease. And I think we all recognize, and the goals of the high throughput approaches is we want to really identify some of the earlier responses. But it's not just, in my opinion, important to identify the early responses. You need to identify the responses that are actually related to these ultimate disease phenomenon. So many of us, and we do a lot of toxicogenomics work as well, we can identify many early responses, but trying to link them to something that's important is, I think, what we really need to keep in mind. I won't repeat this, the so-called toxicity pathways, but I do think it's still important that we need to identify these pathways, or like Chris said, the genes and the targets, and more importantly, we need to identify the interactions between them, which starts becoming a limitation when you start having cell-based systems initially for the discovery phase. We need to determine if, once you identify these targets, identify the chemicals and nanoparticles that perturb them, and again, determine if these perturbations are actually related to an adverse outcome. So I agree we need to pick up the pace. All of us do. But I would like to suggest that high throughput does not necessarily mean high content. For example, and Chris mentioned this as well, if an assay is developed for a specific response, you're really biased for really only asking that particular response. If you have, say you're looking for apoptosis, you'll only get hits or readouts on apoptosis, proliferation, ROS, calcium influx, etc. So we have to recognize that the assays that we put together, inherently the way we construct them limits the information flow. So we kind of take a different approach. Is we thought we would take more of a systems biological approach, and we want to use the early embryonic development. And why do we want to do this? Most would agree that generally during early development, the organisms are generally more responsive to insult. And there's an underlying reason for that. This is the most dynamic life stage of an organism, without question. That the full signaling repertoire is expressed and active at some point during early development. Every gene product is needed to build the organism. So if you're actually hitting these that life stage with chemicals, you have the greatest potential to get an adverse interaction. So again, I refer to this as fewer blind spots because you have this highest potential of interaction. So kind of flipping it around, if a chemical or nanoparticle is developmentally toxic, it must influence the activity of a molecular pathway or a process, and it must hit or influence a so-called toxicity pathway in some way. So in the early embryo, what we do is we want to use the biological response itself to identify the toxicity pathway. And so you got to remember what you're looking at here is you're going to get, the whole organism is not only the signaling needed during development, it's needed at a very precise time in development, and there's relationship between event A and event B and event C. So there's a time and space requirement. So the constraints of development are quite large. So we happen to use zebrafish for many reasons. They share many characteristics with mammals, believe it or not. Molecular signaling is very well conserved across species. With both genome sequence, you can actually identify almost every human gene is in zebrafish at least once. We won't get into that complexity today. We have the technical advantages of cell culture, really, almost all the technical advantages of cell culture, but the power of interrogating a whole organism. It is amenable to rapid and high throughput studies. And again, we want to focus our responses so we get a whole organismal response, and then we can then focus it back down to identify these so-called toxicity pathways. And then once you identify the pathways, in my opinion, this is the best time where you can translate information from these so-called lower organisms back to, say, humans, is really at the mechanism level, not at the endpoint level. And I'll drive that point home in a second. Just a simple reminder of why we like working with zebrafish. We're mostly talking about organogenesis, again, the period of time where most of the organ systems are developing, differentiation, etc. In zebrafish, this is achieved in 48 hours, where you go from a single cell to 48 hours. Most of the organ systems are present and functioning, so it's much more rapid than the mammalian systems. So just so you kind of get your, I'm going to assume that many of you don't know what a zebrafish embryo looks like, but this is what a single cell embryo, single cell newly fertilized egg looks like. So this is 3 minutes after fertilization and the chorion has been removed. So you're looking at the big yolk ball and then the single cell on the top. So you start going through the cleavage stages, and we initiate, for lots of technical reasons, we initiate all of our chemical exposures at 6 hours post-fertilization. So you have the shield stage. So very, very early in embryonic development. And then we for our screening mode, we have a continuous waterborne exposure, no solution changes, sealed plates, so there's no evaporation. But this is what's going on during that period of time. You're going from 6 hours to this organism 5 days later, which is fully formed. Most of the yolk has been absorbed. Here's the eye. So this animal has a fully functioning brain, liver, pancreas, spleen. So this is a real vertebrate. And so we're asked what we're doing. We have waterborne exposure. So these chemicals have the ability to interact and potentially impinge on all of the events necessary to go from here to here. Just so you have an idea what it looks like, we do most of our screening in 384 well plates or 96 well plates. We can go one up on this, but it's really hard to get the embryos in and out of the plates. So you actually look at, if this is a 384 well plate, there's actually 4 embryos. These are 24-hour embryos inside a plate, so they actually fit within a 384 well plate without touching each other. So you put a 5th one in, and they fit nice and snug. And this is what they look like at 23 hours. So in a typical tox screen, so we do drug discovery type screens as well, but in a tox screen, it's not trivial, but it's not really that difficult. So you do need a large colony to produce many eggs a day. So our lab, which is a typical academic lab, we can produce several hundred embryos a day, several hundred thousand embryos a day. And so this has all been streamlined. It's very efficient. We get the eggs soon after fertilization. We remove the chorions. This is an important point. Many labs don't do this. We remove the chorions because, one, we're afraid of that may become a barrier. The chorion itself may be a barrier for some chemicals and certainly for some of the nanoparticles that we work with. But secondly, for the assessments, the image-based assessments that we do later, having the chorion off is dramatically improves the quality of the images. And we put 1 embryo per well in these plates, and we add the test chemicals, and we have a series, and I won't talk about all the different steps of the screening that we do that are done between 1 and 5 days. Some of the endpoints that we look at, so when I say high content endpoints, what I mean is that this is a interaction between multiple events to create an organism, so we can look for things for malformations morphologically. So this is mostly image-based analysis. You can look at a pericardial edema, you see yolk stack edema, body axis curvature, length, width, eye diameter, pigment formation, etc. Circulation, heart rate, developmental progression, all of these can be easily assessed. Right now, many of the students do it by hand, but we've done some automation. I'll talk about that in a second. And we also do behavioral assessments. We ask the question whether or not any of these exposures have impinged on the development of the central nervous system. So we look at spontaneous movements, which is the earliest behavior. We use that endpoint a lot. Frequency of this behavior, touch response, motility, etc. So the point is that what's really nice about the system is that we can control the exposure with precision. So in screening mode, we don't change the window of exposure, but when we're doing mode of action, we could do, say, exposures for just 1 hour in development. Ask the question, if 1 hour developmental exposure leads to an effect, the biology amplifies it. So the requirements to development are so precise, you may hit a pathway very precisely for that very brief moment of time, pull the chemical off, and it derailed the entire developmental program. And it will be displayed by some of these endpoints. And this is what they look like. This is what, at 5 days, what a normal larvae should look like. And this is what an example here is with exposed to dioxin, or TCDD. And so what I'm going to do is show you some of the different endpoints that we very commonly see. This is another example. You'll start seeing some similarities. This is an ethanol exposures. So you start seeing, with increasing concentration of ethanol, more dramatic effects on the embryo. This is some of the nanomaterial that we do, the microinjection studies. Again, you see this, the eye is barely developed, massive, the heart is right here. This is the pericardial cavity, and there's a trunk effect. So all of these can be easily visualized and screened in a plate format. This is another example of a series of compounds that we identified that were developmentally toxic, dithiocarbamates. These animals otherwise look okay, but you can see that their notochord in the normal animals nice and straight, and this one you get this undulated notochord. So this has become an interesting endpoint for us as well. We can also do more, I guess, cellular-based systems in the whole animal. This is looking at cell death in the live animal with an acridine orange assay. So in the normal animal, this is the yolk ball. You can see spots of cells undergoing cell death, and with increasing concentration, in this case with ethanol, we have increased number of cells undergoing cell death. And all of this is amenable to very high throughput screening. We have plate-based assays for this now. We can also do high throughput, and it may not seem high throughput, it is. You can do high throughput in situ hybridization. So you can look for exposures that modulate expression of gene products. And not only, say, quantitative PCR, you can do it that way as well, but you get the spatial distribution of this gene expression. So this we do quite a bit as well. One thing I want to really emphasize is you really have to consider things when you're working with non-mammalian models. You can't get really focused on the endpoints. You have to really think about what caused the effect, not the endpoint itself. So signaling pathways and the molecular events are absolutely conserved between mice and man and fish. But fish are not rodents and are not humans, which was also mentioned earlier. So the consequences of disrupting these conserved pathways can be quite species specific. In other words, a mechanism by which a target is hit is likely conserved, but the consequence may be species or life stage specific. So you can't just say, I mean, sir, we have an endpoint that looks at the fin fold development. We don't have fins, but the molecular pathways that lead to fin fold formation are conserved. So we can use that as a trigger to identify an adverse interaction. So again, these are some of the endpoints that we screen for. And these are really common. So we've screened several 100,000 compounds and nanoparticles at different concentrations, and we see these a lot. That does not mean that these common endpoints are non-specific. What it means is when you perturb development and you have these organisms in an aquatic environment, at the gross level, there's only so many phenotypes that the animal can display. So we kind of take that as an advantage. So if you get a common response of a pericardial edema, you just use that to say it doesn't mean necessarily that the heart's even been hit. It just means that development has been impacted. And so I won't talk about this pathway, just to highlight this effect. So we have the ability to knock out any gene we want in early development in just a couple of days. And so if you have in this pathway, the H receptor and ARNT are present, you expose zebrafish embryos, it leads to embryo mortality within 4 or 5 days. If you knock out either ARNT1 or H receptor 2, bathe these guys in dioxin, the animal is completely normal at this time frame. So this, even though you have these common responses in the pericardial edema, yolk sac edema, reduced growth rate, it has a specific reason. The specific reason is that that pathway, the H receptor pathway, has been hit by a ligand. So you can actually take these animals that have the AHR2 knocked out, now expose them to ethanol, and you'll see the same endpoints. So now this is where the kind of the challenges are. What do you do with this data? There's absolutely no doubt that we can collect billions of data points using whole animal assays. So the question is, for this audience, should we? And what are we going to do with the information once we get it? So we've been struggling with this, and I could definitely use some help as well, but informatically. So again, you're looking at the whole animal, you're getting this, and if you're looking at an exposed animal, you get all of these endpoints. So we're finding ways to extract this information. And how do you summarize it? How do you summarize the data from a whole animal response in some meaningful way that you can make comparisons not only between chemical and zebrafish, but between zebrafish and rodents and humans? So one approach that we've done is we kind of did a hierarchy, a ranking of the effects that we can tease out. And then we give a score. So say mortality would get a score of 1. You multiply that by the number of animals, that would be a score of 24. That would be the most toxic compound. So we call this the easy metric. There's some challenges with this model, but it actually does work. So a top score is 24. With increasing concentration of these series of nanoparticles, you can have some materials that are not at all toxic and others that are highly toxic or have a high easy metric score. Another approach is kind of what Chris mentioned, that we actually do this binary approach sometimes over concentration ranges. You can basically, if you really use the model as advertised, that it's a way to identify adverse interactions, then any adverse interaction is a hit. So in a tox screen, anything that's not normal is something you want to detect. So you can have an animal that has a single endpoint. So say the swim bladder isn't inflated, or an animal that has every possible endpoint, and they get the same score. And then you do that in a concentration range, and then you can compare the EC value, say the EC25 to EC50 versus the NOEL, and use that as a way to identify hazard. We've automated all the image acquisition has been fully automated. So by putting more of these in parallel, we could increase that speed. We've had many other automations that we've accomplished in the last few months. Embryo production, we have no problems in that area anymore. Embryo handling, we've automated the deposition of embryos into the plates, which is a good advance. Microinjection in conditions where you have to deliver the compound directly to the embryo, that's been automated. We have plate-based assays that you can detect some of these cellular-based effects or morphology. And then we also have some automation with behavioral assays as well. So making the data accessible and useful, I definitely want to learn from this crowd how we can actually do that. We need to plug our models into some of the existing really nice tools that we just saw from a couple talks ago. We need widely used knowledge bases, open access. We need new methods for informatics in this whole animal world, and we actually have developed one for the nano base, which has a very similar structure, but it's right now it's focused on nano. Take-home message, I think we all agree no single model is sufficient at this time to identify hazard. I think we all agree with that. I do think that this model and Drosophila and C. elegans as non-mammalian models offer a number of powerful advantages, but interpreting false negatives or false positive, there will be false positive, there will be false negatives, there's no doubt about it. And the easiest one, which came up already today, the difference in the ADME, the metabolism, is clearly an issue. In this model, we have no maternal contribution during exposure. So if you're thinking of developmental tox, that is something to consider. Molecular signaling, again, is functionally well conserved, but the primary sequence not necessarily conserved. And just the last point to make that is, if you go from the gene level, really zoom in, and this is one of the gene targets that we studied, HDAC1. You compare the human, the zebrafish to the human on top, they're 91% similar at the amino acid level. Functionally, these guys are very similar. We have studies to prove that. So that makes sense. There's some reassurances there. So if you have a chemical that inhibits an HDAC in humans, it's probably going to inhibit the HDAC in zebrafish. You come in another example, TGF-beta1, which is functionally very well conserved in zebrafish and also with humans, but the identity is only 44% with 61% similarity. So this is another possibility how you can have a false negative or a false positive, because the actual target of a chemical may interact with these species in a different way. All right, thank you.
Chris Borgia from NIEHS. We've done a lot of work with C. elegans. In fact, we just had a series of papers come out in PLS1, PLOS1, dealing with the actual analysis and interpretation. And in those papers, we talk about difficulties in doing the high throughput screening type activities there, mostly in handling cylindrical animals,
Right.
as compared to handling, when you're plating, the little the eggs, which are nice and round and
Right.
easy to work with. Do you ever pick up the fish out of the well and run them through a sampler of some sort for some of the screening? Do you have problems in doing that, like we see with C. elegans?
Yeah,
Some of the difficulties?
so I know John, he uses, the COPUS instrument from,
Right.
and we don't use that instrument. That was problematic for us as well. We actually use robotics to move, very similar to the robotics that Chris showed, to move the embryos from place to place. But most of the, once we move it into the well, we do the exposure there, and we do the assessments in that well as well. So it's a microscope-based image. It's not a flow. That would be, I think, the new advance that's not available yet. To have something that can take an animal in flow, like a cell sorter, and analyze it on its way through in 3 dimensions, multiple cameras. I think that would be a nice advance. But,
And one other question. Why'd you go from 4 embryos per well to 1?
yeah, that's a great question. So we go 1 per well because you have the influence. If there's a mortality, say you're in the middle of the curve, if you have mortality of 1 embryo in a well, it actually impacts the other organisms next to it. So it really reduces the throughput. Even though you think you're doing multiple sampling from 1 well, it actually hurts you in the end. So I'd rather do replicate plates. So typically we'll do 1 compound per well, and then we'll stack the plates. So we can do 96 compounds at a time, and we replicate it up rather than laterally.
We're finding now an incredible well effect in the C. elegans that we're at a loss to explain. And so you made a wise choice, I think.
Yeah. Okay.
Risha Putzrath, EPA. Just a quick comment on your EASY score, which is to be careful about combining hierarchical and potency. And if you want a complete discussion of that, in the 70s and 80s, we did it on cancer potency with strength of evidence.
Right.
The compound is carcinogenic, and all the pitfalls are well
Right. Yeah, I understand.
enumerated.
Yeah, I completely understand. Thanks.